So let's start with telophase now. Telos means end. Telos means end. Phase means stage. So this is the last stage of karyokinesis of mitosis. Last stage of karyokinesis of mitosis. So let's see what happens during this telophase of mitosis inside the cell. So in this telophase, actually by the end of anaphase, all the chromosomes or chromatids of the chromosomes have reached towards two opposite poles of the cell. Then after reaching, that is in the early telophase, these chromosomes start undergoing decondensation. So these chromosomes undergo decondensation into chromosomes, or we can write chromatids. Chromosome with one chromatid is also called as chromosome, or a chromosome having two chromatids is also called as chromosome. So chromosomes undergo decondensation. Decondensation into chromatin fibers. Decondensation into chromatin fibers. Chromatin fibers. That means chromosomes have lost their identity. No more they will appear as chromosomes. So let's see how they undergo decondensation. So this is the cell membrane of animal cell, and here what is present? Centrosome is present. Here one centrosome, and here centrioles are present in each centrosome, which are arranged perpendicular to one another. Now the chromosomes which have reached towards two opposite poles of the cell have lost their identity due to decondensation into chromatin fibers. Here also these chromosomes have lost their identity and decondensed into chromatin fibers. Okay, so nucleolus, which got disappeared, is reappearing during the telophase. So here also nucleolus reappears, and nuclear membrane around this chromatin fibers, that is genetic material, DNA, is reappearing. This is nuclear membrane. So by the end of telophase, one nucleus of the parent cell becomes two daughter nuclei. Next change. So what is the next change? All other cell organelles. All cell organelles like Golgi bodies, then ER, endoplasmic reticulum, and all other cell organelles which have disappeared during prophase, that is by the late prophase, will reappear during this telophase. So telophase is completely opposite to prophase. So whatever the cell organelles got disappeared during prophase, that is by the end of prophase, reappear or reform in the telophase. So all these cell organelles will reform. Next. So the spindle fibers, microtubules present in the spindle fiber undergo depolymerization, and hence the spindle fibers of the spindle apparatus also disintegrates. So these are all the changes that occur during telophase. Next, this telophase is followed by cytokinesis. Now let's study cytokinesis. So cytokinesis is nothing but cytoplasmic division. So actually we started with mitosis. In this, mitosis occurs by nuclear division, that is karyokinesis, and cytoplasmic division, that is cytokinesis. Under nuclear division, we studied about all different phases of nuclear division. Karyokinesis, that is prophase, metaphase, anaphase and telophase, which is followed by cytoplasmic division now, that is cytokinesis. So cytokinesis occurs in both plant and animal cells. In case of animal cells, this cytokinesis occurs by Furrow method. Cytokinesis occurs by Furrow method. Furrow method, by the formation of furrow in the cell membrane in case of animal cells. In case of plant cells, the cytokinesis occurs by the formation of cell plate. Cell plate at the center of the cell. So this method is called as cell plate method of cytokinesis, occurs in plant cells. plants. Okay, now let's start with the cytokinesis of animal cells, which occur by Furrow method. So, in case of animal cells, this cytokinesis occurs with the help of microfilaments, MF. They are microfilaments. Let's see how this cytokinesis occurs in animal cells by Furrow method. So let's say this is the animal cell. Animal cell lacks cell wall. So this is the cell membrane, which is the limiting boundary of animal cell. So here nucleus is present. Inside the nucleus, genetic material, that is genome, DNA, is present in the form of chromatin fibers. And this is nucleolus. Here also another nucleus is present. Genetic material is present in the form of chromatin fibers, nucleolus. And here what is present? Centrosome, having 2 centrioles. Here also centrosome is present, which is with 2 centrioles. Centrosome is present, which is with 2 centrioles here also. Okay, now let's study the cytokinesis which occurs in animal cells by Furrow method. So this occurs with the help of microfilaments. So these microfilaments are aligned. They are just accumulated. These microfilaments are accumulated just below the cell membrane and at the equator. Microfilaments are accumulated below the cell membrane and at the equator. Equator means that is at the center of the cell. These microfilaments, after accumulation, they are forming a ring-like structure. This ring-like structure is called as contractile rings. So what is happening? Contractile rings are formed just below the cell membrane, that is at the equator, due to the accumulation of microfilaments. So these are called contractile rings because they are contractile in nature. These rings made of microfilaments are contractile in nature. They can contract. So as they can contract, they pull this cell membrane towards the center of the cell from periphery. Let's see, in this diagram I will be explaining. So they pull this cell membrane. In this diagram I am explaining. So they are pulling this cell membrane as these contractile rings made of microfilaments are contracting. They pull this cell membrane towards center from periphery. And this cell membrane is pulled towards the periphery, towards the center from periphery, and it extends deeper into the center of the cell. It extends. So this is called furrow now. This is called furrow. Where this furrow is formed? This furrow is formed in the cell membrane, that is towards periphery at the equator. And this furrow which is formed is deepened, and both of these furrows are meeting at the center of the cell, dividing the cell into 2 daughter cells. So this is the way how cytokinesis or cytoplasmic division occurs in animal cells. So it occurs with the help of microfilaments. These microfilaments are contractile in nature. They are aligning at the just below the cell membrane and at the equator. After alignment, they are forming ring-like structures called contractile rings. These are called contractile rings because they are contractile in nature. So as they are contractile in nature, they are pulling this cell membrane towards the center of the cell, and hence furrow is formed in the cell membrane towards periphery and at the equator. Both of these furrows will deepen into the cell and they divide the cell into 2 daughter cells. So this is about the cytokinesis in animal cells. Next, let's study about the cytokinesis in plant cells.